Assalamualaikum, good afternoon. We are from Group A. Today we were gonna talk about bacteria causing disease in plants. Before we start, let me introduce our member. First, me, Gracela Nanda, with student number 169, Safira Tasya with student number 171, Akhmad Rudy with student number 172, Setyawan with student number 173, and Coryn Adelina with student number 186. Okay, our topics one, introduction, uh, bacterial disease caused by bacteria, and candidate genus against bacterial pathogens. Next. Biotechnology is defined as a set biological techniques developed through basic research. In other words, it is the genetic manipulation and multiplication of any living organism resulting in the production of improved and new organisms, and products can be used in a variety of ways. New biotechnological tools enhance pathologists' ability to make improvements in crops regarding to their respective diseases. For example, it can be applied as genetic engineering. New techniques in plant genetic engineering for the management of plant diseases are now available, and the application of genetic engineering for plant disease resistance. Hence, plant genes and their products that interact with plant pathogens have been identified and either inserted into plants to improve plant breeding for disease resistance.
So next is bacterial diseases. So detection of pathogenic bacteria in seed and other plant tissue is challenging because the target bacteria are often irregularly distributed and present as a small component of a much larger bacterial population. The second one is a new and modern molecular techniques, nucleic acid-based technique, have been demonstrated to be generally more sensitive, specific, and reliable of bacterial plant pathogens than other methods. Among of this detection technique, PCR assay has been used very widely. A PCR-based assay using the primers from DNA sequences of the holotoxin gene was able to detect several pathogens efficiently. Next.
The appearance of bacterial grow in our plant tissues indicates bacterial plant disease may be present. There are uncommon microorganisms called molecules. These microorganisms are very small where they must be examined by electron microscope. These microorganisms cause severe plant disease such as plant stunting, yellowing, or redding of leaves, production of abnormal flowers, and eventual decline and death of the plant. Molecules can be diagnosed by several parameters, such as symptom determination, grafting, transformation, microscopical examination, and susceptibility to tetralation.
Okay, the next one is candidate gains against bacterial pathogens. So this is one of the example of candidate gains. A wide spectrum bacterial-like resistant gains called XA21 sourced from an African rice was backroasted into cultivated rice plants in Philippines. This resistant genes was cloned using molecular means and distributed to all over the world so that the gene could be put into rice cultivars of local importance.
Okay, from this topic, we can conclude that plant pathogenic bacteria cause many different kinds of symptoms that include galls and overgrowths, wilts, leaf spots, spike and blades, soft roots, as well as caps and chancres. Bacteria are spread in many ways. They can be splashed about by rain or carried by the wind, birds, or insects. Bacteria require a wound or natural opening, such as stomata, to get inside a plant host, and once inside they then kill host cells. Uh, this is the reference that we use to make this presentation. That's all our presentation. Thank you.